These therapies did not exist 20 years ago. And now we see people who had weeks left to live, totally cured from their genetic diseases.
That combination of a super interesting problem and a super passionate community, that's why I do what I do.
So when I started, gene therapy was at a different place. So my PhD started about, let's say, 15 years ago. And well, back then we didn't have a lot of examples that would show that gene therapy works. Actually, there had been a few setbacks. And so people weren't altogether super enthusiastic about the method until Jean Bennett came around.
Did you know about that blind dog?
No.
So it's called a Breyer dog. So it's a breed that occasionally has a mutation in this, like the counterpart of the RP65 gene. So if they are double negative, then they are blind.
She tried gene therapy using AAV, so the same type of virus that we use today, and she cured dogs from blindness. And so that was quite fascinating back then. And so this continued being tried in kids as well.
Our clients are really longing for a process to make viral vectors for gene therapies that is efficient, scalable, and has reduced costs, because ultimately that cost is passed on to the patients. And so as Cytiva, we try to help our clients in all the steps within that journey to achieve those objectives. And cell line is one of the most important ones, because cell line or cells is actually where the viral vectors are manufactured. So having a cell line that really is fit for purpose and shows good process performance is really key, and this is what the team in Cologne is busy with every day trying to achieve.
Cytiva Cologne was founded 2001, so a long time ago. It was a spin-off from the University of Cologne. We have now 20 years of experience of cell line development, and all these challenges we have in the past made us expert in cell line development. When you think about cell line development or the history of cell line development, it's what we see now in the AAV field. It was the same in the monoclonal antibody field, right? So they started to just using the hybridoma cells and then see, okay, actually for a larger scale up, it's not usable because you need really a lot of antibody during the clinical trials in the clinical applications. And then they started with a short cell line, actually, also with a transient transfection, and then ended up, as we do, with a stable system.
Yeah.
That's quite cool.
You know, when I started with this company, I was very skeptical about stable AAV producer cells, because when you think about it, replicating AAV as a virus has zero interest in having a cell survive that it lives in. And then also cells are very good at detecting viral infections. There's literally like millions of years of experience with both of them fighting each other. And so it was the sheer idea of having, taming the beast that is AAV and having it stably integrated in a cell. I think many people thought it was impossible. And so people were not super, buying into that idea until, Cytiva Cologne came around. Back then we were called CEVEC. And so they found this incredible method. Like, this was really this combination of this in-depth knowledge about how to do cell line development in general, like how to put in a gene into a cell and then select it.
To produce an AAV particle, you need a lot of components, and all these components are normally toxic to the cells. So the cells, if these elements are expressed in the cells will die. And we found a way to make all these components inducible. That means you just add a component to your cells, and this component then leads to the expression of all the components you need for the AAV production. And that helps that the cells will not die, and you can produce your AAV.
Because stable producer cell lines really drive the manufacturing costs down. And this is really important for gene therapies specifically, because they're aimed at sometimes rare genetic diseases where the patient population is quite low, which means that the manufacturing costs and the R&D costs are spread amongst just a few patients sometimes. But equally, even for the larger indications where the patient population is bigger, these gene therapies are really changing their lives. This is not a treatment, this is a cure.
And that's also the reason why I decided to be a scientist, is to understand the biology, what happens in the cell, what happens in the small viral particle, and what leads that this small viruses can infect so many people, for example. But on the other hand, using this biology, and if we have now these tools with this biology knowledge to treat people, to help people to survive diseases that would not be or is not treatable before, of course, that is something that makes me proud a little bit. Yeah.
And it's something quite brutal for the patients, you know, to know that you were born with some genetic mistake that you have no control of, and you did nothing wrong to get it. And that's the destiny that was describing how your life is going to go, and suddenly there is a cure for this. So I think it's just amazing what humans are able to achieve and what the science can deliver to us.